So good evening, everyone. Welcome to the webinar tonight, and thank you for joining. I'd just like to start by thanking Tim Elliott and the team at Virbac for providing the opportunity for me to talk to you tonight about a topic that I'm very passionate about, being animal health. So tonight we'll be talking about vaccination of sheep and cattle specifically, and hopefully we can run through some principles and best practice scenarios as we go through. So I just thought I'd start by telling you a little bit about me. So obviously already been introduced, but my background is largely in research. I've worked with CSIRO for over 30 years now in the livestock research space, with a particular focus on animal health and particularly on ruminant immunology. And my focus there has really been on developing strategies to help livestock producers reduce reliance on antibiotics to prevent and treat disease. As livestock producers, we all aim to minimise our use of antibiotics in our animals, but of course, at times we need to use antibiotics from a welfare perspective. So a lot of my work is focused on providing producers with tools to help them achieve both a reduced reliance on antibiotics, but at the same time maintaining animal welfare. And a particular focus has been around assessing responses to vaccination and general immune system stimulation. So 30 years working for CSIRO. I guess a couple of years ago, I probably had what you call a midlife crisis, where I decided I'd like to explore a couple of other avenues and started working on a part-time basis as a livestock agent. As part of that role, I probably do more and more around the livestock consulting, specifically in areas of animal health and genetics and genomics, which I'm very passionate about. So I just thought I'd quickly run through what we will talk about tonight. So we'll start by just having a quick recap of the immune system and how it works, and then talk a little bit about what we're actually trying to achieve using vaccination in our animals. We'll talk about the types of different vaccines that are out there available for commercial livestock producers and the pros and cons of each of those types of vaccines. A little bit about how the vaccines work and how they induce responses in animals. We'll talk about vaccination protocols and particularly why boosting of animals is important. We'll also talk a little bit about the role of maternal antibody in livestock, the important role that those maternal antibodies play when passively transferred to calves and lambs. Then we'll talk about factors influencing vaccine efficacy, and that's a really important topic, I think, to cover tonight is what are those factors that can impact the efficacy of the vaccines we have available. And then we'll spend some time talking specifically about best practice vaccination techniques. So just a very quick basic immunology recap. So we're likely all familiar with what the function of the immune system is, and that's to really protect us against pathogens. So I'll use the word or the term pathogens a lot through the webinar tonight, and when I'm talking about a pathogen, I'm really talking about any type of organism that can cause disease. So when I talk about a pathogen, that's what I mean. So we often think about our immune system in terms of just protecting us against pathogens, but it also plays an important role in other areas. And one of those is it plays an important role in removing worn out cells and repairing tissue, especially following injury. But it also plays a very important role in protecting us against the development of cancer. And that's through immune surveillance and picking up those cells that have become cancerous and basically eliminating them from the body. So the immune system has multiple functions beyond just protecting us against pathogens. So when we think about how the immune system operates, if you like, there's really two arms to the immune system. One is what we call innate immune defense and one is adaptive or acquired immune defense. So the innate part of our immune system is really designed as the first port of call in defense against a pathogen. So innate responses are very fast, so they're initiated very quickly, and the idea with innate type responses is really to very quickly get that infection, stop replication of bacteria or viruses in the system or whatever the pathogen might be, and to contain that infection. So the innate immune system is triggered into action very quickly, happens very fast. It's largely non-specific, which is really important because first time our immune system sees a pathogen, it's really important that we can respond to that pathogen quickly. And often we haven't seen that pathogen before, and therefore the immune system, the innate immune system, functions from a largely non-specific point of view, trying to get things under control very quickly. And another characteristic of the innate immune system is there is no real memory component. And what I mean by that is once an innate response is induced in the body, the next time we see that pathogen, the innate response doesn't necessarily get better or learn from that response, which is quite different to the adaptive or acquired response. So you'll see in our diagram there, we've got innate and then the other arm of the immune system really around adaptive and acquired. So the adaptive and acquired system, it's quite slow to generate responses, and part of that is because it's very highly specific. And so it takes time for adaptive and acquired responses to develop in the body. But what's really important about adaptive and acquired responses is they generate a memory component. And that's really important when we're thinking about vaccination. We want to generate that memory component. So we can further divide that adaptive response into what we call cell-mediated and antibody-mediated responses. So we often talk about antibodies when we talk about responses to vaccination, and they're very important, of course, but we often don't talk about cell-mediated responses, which are also very important. So vaccination really attempts to trigger both types of responses, and depending on the pathogen, if it's an intracellular pathogen, so we're thinking of things like viruses which enter your cells, then cell-mediated type immune responses are really important for controlling those type of pathogens. Whereas soluble type antigens or toxins, for example, that are extracellular, so they're not necessarily inside your cells, then antibody-mediated responses are really important to control those type of pathogens. So really the aim of vaccination is to expose the immune system to a pathogen, either the whole pathogen or parts of that pathogen, to induce an immune response in the animal, which has a memory component. And it's really, the memory component's really important because we want the immune system to remember what it's seen, so that if it sees that same pathogen in the future, it will respond very quickly and very specifically and in an attempt to get that pathogen under control. So we all heard lots about vaccination during COVID, and many of you would have been vaccinated during the years where COVID was really at high infection rates, and that is obviously in an attempt to reduce the spread and the severity of the disease. So if we think about vaccination, what we're really trying to target with our vaccination is induction of strong adaptive and acquired responses, and it depends on the type of pathogen which of those type of responses will be most beneficial in helping protect against those pathogens. So you'll also hear me talk a lot about antigens and antibodies throughout the webinar tonight, and the picture we see here, the Y-shaped molecule, is an antibody, and we've probably all heard about antibodies and understand to some extent what they do. But antibodies, their primary function is to bind antigens. So you can see in the diagram there that on this Y-shaped antibody, on the ends of those variable arms, there are two spots, one on each side of the antibody where it can bind antigen. And the antigens that we include in our vaccines are antigens from the pathogens we're trying to protect those animals against. So antigens, again, can be part of the pathogen itself, or it can be something on a whole pathogen. But the important thing is those antibodies can bind those antigens, and that is that goes a long way towards helping us or the immune system protecting us against that pathogen. So you just think of antibodies and antigens. Antigens are the things that antibodies bind to in our vaccines. So I just thought I'd run through a few different types of vaccines. And this list is by no means extensive. There are several other types, but these are the main types of vaccines. And how the vaccines generally differ is in the type of antigen that is in the vaccine. So in a live attenuated vaccine, these type of vaccines actually contain live pathogens, so live bacteria or viruses, but they've been attenuated or weakened, which is really important because when we vaccinate with live attenuated vaccines, we want these pathogens to replicate but not to cause disease. So these are live pathogens, but they've been attenuated or weakened in some way. There's various different ways that we achieve that when we generate our vaccines, but they're generally very effective vaccines because they're very close to what the real pathogen is that the immune system would see. The next type we'll talk about are inactivated vaccines, and these are where they are again whole pathogens. So you can think of a whole bacteria, if you like, but they're inactive and they've been killed. So this means that they generally can't replicate, but the immune system still gets to see the whole pathogen. Next type of vaccines we'll talk about are the subunit vaccines, and these are the vaccines where the antigen is a piece of the pathogen, not the whole organism. So we've gone through from a live attenuated where we have a whole pathogen that's live to inactivated vaccines where again the pathogens are whole pathogens but they're killed, to subunit vaccines where the pathogen in this case, or the antigen, is a piece of the pathogen, not the whole organism. So we'll obviously talk a lot about clostridial vaccines today because clostridial vaccines are very commonly used by most livestock producers. And so in our clostridial vaccines, many of these contain are toxoid vaccines that contain inactivated toxins. So for many of for example, the clostridial bacteria, it's actually the toxins that the bacteria produce that are harmful to the animal rather than the bacteria themselves. So what we're trying to protect the animals against in these type of diseases are the toxins they produce. So a toxoid is simply an inactivated toxin. So in toxoid vaccines, what we are vaccinating with is antigens. The antigen in those vaccines is a toxoid or an inactivated toxin from these bacteria. So in all four of these cases that we've talked about here, these are all antigens that we are delivering via the vaccine, the antigen itself. In the last two examples, these are examples where we're using vaccines to deliver instructions to the animal's own cells to produce the antigen for us in the system. So viral vector vaccines, this is where we use a harmless virus to deliver the genetic code or DNA which instructs the animal's host cells to actually produce the protein antigen for us. And as part of that process, that antigen is produced by the host cells, and then it is detected and presented to immune cells. So in this circumstance, rather than us actually delivering the antigen itself, we're actually delivering with our vaccine the instructions, if you like, for the host cells to produce the antigen for us. And just a little recap, that our DNA, so we all have DNA, and DNA is transcribed into what we call messenger RNA, and that is what instructs how to produce the proteins in our body. So mRNA vaccines, which you would have heard a lot about during around COVID, in these type of vaccines, they contain small pieces of messenger RNA, which again instruct the host cell to produce the antigen for us and present it to immune cells. So these are a couple of the more modern technology, vaccine technologies, I guess, where we're actually using the animal's own cells to produce the antigen for us. So there's pros and cons with all the different types of vaccines, and I'll just concentrate here on the live attenuated, the inactivated, and the subunit vaccines, and talk about a few specific characteristics of the vaccine. So generally, live attenuated vaccines produce a very strong response in the animal, and that's because the antigen in those vaccines, being a live bacteria, for example, is very close to what the immune system would see in an actual infection scenario. So live attenuated vaccines are very effective at inducing a very strong immune response in the animal. And as we move, if you like, further away from what the immune system would see, the ability of those vaccines to induce an immune response in the animal is reduced. So generally, live attenuated vaccines will produce a stronger response in an animal than an inactivated vaccine, which will produce a stronger response than a subunit vaccine. As a consequence of the strength of that response that's induced, we also get a corresponding increase in the length of protection. So generally, and I'm talking in general terms here, there are situations where this doesn't apply, but in general, live attenuated vaccines will also give you a longer length of protection, and that's because of the strength of the immune response they induce in the animal. And again, we see that length of protection generally is reduced as we go from a live attenuated vaccine to an inactivated vaccine to a subunit vaccine. So of course, with pros, there's always some cons. So one of the cons with using live attenuated vaccines is the stability of the vaccine. So because it is a live vaccine, stability is something we have to be really careful with, the stability of live attenuated vaccines. And we'll talk more about vaccine stability later in the webinar. But just be aware that those live attenuated vaccines are generally less stable than something like a subunit vaccine. So another con we can think about is actually safety for operator and recipient. So as an operator, we need to be much more careful using live attenuated vaccines than we do, for example, than using a subunit vaccine. And not only safety for the operator, but it's also important to consider the safety of the recipient. So generally, when we give a live attenuated vaccine to a healthy animal, that pathogen, that attenuated pathogen, can reproduce, but it won't cause disease or infection. But if we give that live attenuated vaccine to an immune compromised animal, sometimes that can lead to disease. So we just have to be a little bit more careful with those types of vaccines. So as I mentioned, we'll talk more about clostridial vaccines than the other vaccines tonight, because we're probably most familiar with those type of vaccines. And here's an example of the Webster's 5-in-1 vaccine. I've just pulled this information off the Virbac website. You'll see, obviously, we're protecting against 5 Clostridium bacterial species here. And you can see from the table at the bottom that for 4 of those Clostridium species, it's actually the toxoid that is in the vaccine. So remember, the toxoid is the inactivated toxin. But for the Clostridium that causes blackleg, so it's actually the bacteria, the whole bacteria that is in the vaccine. So the Webster's 5-in-1 vaccine, similar to many of the 5-in-1 vaccines on the market, is what we call a bacterin toxoid combination. So it contains both bacterin, which are killed or inactivated bacteria, and it contains toxoids, which are the inactivated bacterial toxins. So if we went back to our previous slide, then we would say that these are a combination between a toxoid, so a toxoid vaccine, and an inactivated vaccine, so a bacterian toxoid. So most of the clostridial vaccines are generally all bacterin toxoid combinations. And there's lots of different vaccines on the market for different diseases, and you'll see various different types. So I just wanted to talk a little bit about adjuvants. So we've talked a lot about antigens. So antigens are the things in those vaccines that we want the immune system to see. Adjuvants help the immune system to see those things. So I always use a bit of an analogy for adjuvants to explain to people how they work. And that is, when I was a younger person going to the pub, I was never a good-looking rooster, but I had a mate who was a really good-looking rooster. So I used to hang around with him at the pub a little bit. And so if you think of him as the adjuvant and me as the antigen, and potentially the girls at the pub as the immune system, we're starting to see how this unfolds. So the immune system, or the girls, are attracted to the good-looking guy who's the adjuvant, but as part of that, they also see me as the antigen. So I like to use that bit of an analogy to explain how adjuvants work. So in many cases, the antigens in our vaccines, if we didn't have adjuvants in those vaccines, our immune system would not respond strongly to those antigens. It's actually the adjuvants that are helping us achieve that in our vaccine. So not all vaccines require adjuvants, but many do, and they're added to improve vaccine efficacy. And what I mean by vaccine efficacy is the ability for those vaccines to protect you against disease. So they're especially important in vaccines that contain poorly antigenic components. And what I mean by that are antigens which the immune system doesn't take a lot of notice of usually, but with an adjuvant present, it helps bring that immune system to the party and look at that antigen and respond. So the adjuvants trigger innate immune responses that enhance that adaptive response induced by vaccination. So that's really important. The other thing we can use adjuvants for is actually to shift the nature of the immune response towards a predominantly antibody or cell-mediated response. So if we know for a particular disease that, for example, a cell-mediated response will be more effective, we can use adjuvants that effectively push the immune system more predominantly down that track. The other one that adjuvants are really important for is often they reduce the dose of antigen required in a vaccine to achieve protection. So that helps us reduce the cost of vaccines when we're developing them. But the really important factor that adjuvants help with is prolonging that immunologic memory. So how long do we remember those antigens that the immune system's seen? Adjuvants are very important in extending that period of memory. So in a typical clostridial vaccine, you'll see here under the composition label, which I've just taken off the Virbac website, that it says that they're aluminium adjuvanted. And for most of the clostridial vaccines, they'll contain an aluminium salt, and that is what is acting as the adjuvant. But there is a whole lot of different agents can be used as adjuvants, and I've stuck some of the, you can see some of the more common ones in this diagram here on the left. But up the top there, you can see mineral compounds like the like aluminium hydroxide. Some of the aluminium salts are very common adjuvants, and especially common for clostridial type vaccines. So how do vaccines induce immune responses? Now, this is a very busy diagram, so I don't want you to get too concerned about the content here. But I'd just also like to point out here we're specifically talking about inducing antibody responses, but remember those cellular responses are important as well. So if you look up on the top left, we see these specialized cells in our immune system, and here we've got a dendritic cell. You can see that this dendritic cell has seen an antigen, and this would be an antigen delivered, for example, by your vaccine. In this case, it's a bacterium, and it will basically what we call phagocytose or engulf that bacterium. And think about dendritic cells a bit like the old Pac-Man game where, you know, these things come and gobble things up. That's exactly what these type of antigen presenting cells do. They gobble things up. And you'll see what these cells are really good at doing is taking these antigens and degrading them, but then presenting them to cells of the immune system. So you can see there how that antigen presenting cell is actually presenting that antigen to a T cell. Now, we're not going to get into T and B cells. They're lymphocytes. They're very important immune cells. But that T cell recognizes that antigen, and those helper T cells are what go on to help B cells in our system produce antibodies. So the B cells or the B lymphocytes in our body, they're the immune cells that pump antibodies out. So if we go to the next step, we can see on the bottom left of the diagram there where that helper T cells talking to one of your B cells and basically showing it this the B cells presenting this antigen to the helper T cell, and it's providing help to tell that B cell what it needs to do. Now, the diagram on the right is a really important one where we've seen a B cell that's been activated. These will generally go on to either produce antibodies. So you can see those cells that are labeled as plasma cells. They're the ones producing antibody. And these cells are really like an antibody factory. So they produce huge amounts of antibody and pump that antibody out. And that is the antibody that we see floating around in our circulation or at mucosal surfaces. But the important other function of these B cells is we get these memory B cells produced, and they hang around for a long time. And what they're really important for is if we see that pathogen again or that antigen again, they remember that antigen and they respond very quickly to tell these B cells to produce more antibody. So that memory component is really important for the success of our vaccination protocols. So I just wanted to talk a bit about boost vaccinations and why we need them. This diagram specifically for clostridial vaccines, and this is a very typical, if you like, antibody time course you would see for a clostridial vaccine. So you can see the sensitizing dose. So what we mean by that is that's the first vaccination the animals received. The important things to note there when we look at that response in green is, firstly, it takes a little while to develop a level which we would say the antibody level in the system is protective for the animal. So the red dashed line across there is a theoretical protection level. So when antibody levels are above that dash line, we would expect the animal to be protected from the disease. When the antibody levels dip below that protection line, animals will be susceptible to disease. So the real key with our vaccination is to keep our antibody levels above the protection level. So you can see when we give an animal a sensitizing dose that the time we see where the antibody levels are protective are actually it's a fairly short window of just maybe 4 to 6 weeks. After that, the levels of antibody drop off quite quickly to a point where the animal's no longer protected. So if we don't boost an animal, and what we mean by boosting an animal is give them a second vaccination, we need to be giving animals a second vaccination before those protective levels of antibody dip below that line, because otherwise there's a period there where animals are not protected. So as producers, it's very convenient, for example, with, say, weaner calves, to vaccinate at marking, but then we often don't give them a boost till weaning. Now, sometimes that window could be up to, maybe 4 to 6 months. So you can see pretty clearly from this diagram that after, say, 4 to 6 weeks from that sensitizing dose, those animals are actually at risk of disease after that period. So certainly if you're marking calves and giving your first clostridial and then not giving a booster till 4 to 6 months later at weaning, there's a big window there where those animals aren't protected. The other thing that's really important to consider is if we boost vaccinate while the antibody levels are relatively high, those antibody levels we achieve with that boost vaccination will be much higher than if we let the antibody levels really drop dramatically. So, and the consequence of that is we'll get a longer period of protection from that second dose or booster vaccination. So that's why we need to give boosters. Why does the frequency of required boosting vary between vaccines? And it's largely to do with the type of vaccine. So we have some vaccines, and Guadair is a good example, where single shot lifetime protection. Now, the reason it does give lifetime protection is, one, the type of response it induces in the animal, but also the strength of that response it induces in the animal. So we get a whole range of different requirements for different vaccines. So if anyone out there is familiar with the Virbac vaccine for Barber's poleworm, it actually only protects for a period of 6 weeks, requiring another dose. For most of the components of a clostridial vaccine, once we've primed animals, that second dose will protect them for a year. But it's also important to note that that's not the case for all components of that clostridial vaccine. So, for example, if you want to protect animals against pulpy kidney, we know you need to be vaccinating more frequently than annually, and potentially every 3 months during those high-risk periods. So, what is the optimal time to boost? The optimal time to boost is while there's still sufficient antibody in the system that we're above that protection level. What's the consequence of not boosting in the suggested time frame? Is that we have a period where animals are unprotected, is one consequence. The other consequence is the antibody levels we achieve with that next dose will not be as high if we're outside that period. So maternal antibodies, I just wanted to talk quickly about maternal antibodies, why they're important. So as many of you will already know, both calves and lambs are born with no antibody from mum. So there's no transfer of maternal antibody across the placenta in ruminants. So very different to the situation with humans where babies, human babies, they receive maternal antibody across the placenta, so they're born with antibodies from mum. Calves and lambs are not. So they're the first time that they'll receive antibodies is through colostrum. And that highlights the reason why getting colostrum into lambs and calves is really important, because it's critical for them to get those maternal antibodies to protect them until such time as their own immune system can start to protect them. And it's a very small window. So within 48 hours of birth, calves and lambs will not uptake through the intestinal. Basically, what happens is that system closes, and they will no longer take maternal antibody up into the bloodstream by absorbing it through colostrum. So really critical to get that colostrum into lambs and calves. How long do these maternal antibodies provide protection? Now, we know from research that maternal antibodies have a half-life of somewhere between 7 and 21 days, which means between every 7 to 21 days, the amount of maternal antibody in the system is halving. So maternal antibody only protects the animals for a relatively short period of time. And you can see in the graph here, I've attempted to draw a little curve there at the start showing maternal antibody. And, definitely within 10 to 12 weeks of age, there'll be very little maternal antibody in the system, and certainly not at levels that can protect that animal. So how do we maximize transfer of this passive immunity to calves and lambs? And that is by vaccinating your ewes and your cows prior to calving or lambing. And if we provide their annual boosters at that time, what we're trying to achieve is maximum transfer of those maternal antibodies into colostrum, and therefore maximum uptake by those neonates suckling that are receiving those antibodies through colostrum. So to maximize transfer of passive immunity, two things important. Make sure that we're giving our cows and our ewes their annual booster. I like to say a couple of weeks prior to the commencement of lambing or calving is ideal. And also ensuring as best we can that animals are actually getting colostrum. So especially people that raise lambs and calves as potties, it's really important that they get colostrum. So when do young animals need to get their first vaccination? The answer to that is prior to when maternal antibodies are no longer at protection levels. There is evidence that if we vaccinate animals very early, that maternal antibody can actually inhibit responses to vaccinations. But the ideal time to be giving young animals their first vaccination is at that time where maternal antibody protection, the protection afforded by maternal antibodies is starting to be reduced to a point where those animals are no longer protected. Excuse me. So I just thought I'd talk now about factors that influence vaccine efficacy, because we often, we hear about failures of vaccines to protect animals at different times. And in a lot of those circumstances, it's not the fault of the vaccine. So sometimes, obviously, the vaccine itself can influence the efficacy of vaccination. But I rest assured that any vaccines available on the market in Australia for livestock producers have been well tested to have good efficacy against their targets. And for anyone to commercially sell vaccines in Australia, APVMA, so that's Australian Pesticide and Veterinary Medicines Authority, have very strict guidelines on the evidence you need to provide to show that vaccine is efficacious before they'll allow you to sell it to producers. So more often than not, failure of vaccination is actually due to things other than the vaccine itself. So we'll talk specifically about a couple of factors that influence vaccine efficacy, and by no means is this this is an exhaustive list, but these are a couple of the key ones I see often. And one of those is stress. We know ourselves when we're stressed, that's when we become sick, our immune system compromised. Sorry, voice is giving way. The other one is nutrition, and we'll talk about macronutrients and micronutrients. And the other important one is genetics, actual genetics of the animal. So we'll just run through a few of these. So stress. So when we encounter a stressor, and that can be physical or psychological, it initiates a cascade of events in our body. Ultimately, that leads to the release of cortisol. So you've probably heard about cortisol before, hormone in the body that has several vital functions in our body. But one of the key ones is actually modulating the immune system. So when we encounter a stressor or in a stressful situation, cortisol is released. Elevated cortisol levels have been shown to suppress almost every aspect of the inflammatory response. So they have a major impact on immune system function. And there's lots of evidence showing that it can impair, that elevated cortisol can impair lymphocyte function and reduce antibody production. So you can see some of the new products out there that have chromium in them, and certainly Virbac have a product with chromium. And chromium has been shown to be very effective at reducing cortisol levels. And by lowering those cortisol levels, they can help us maintain a stronger immune system. But it's probably no surprise, we often get sick when we're stressed, and that because when we're under stress, our immune system is very much compromised. And it's no different when we're talking about livestock. But I just wanted to flag a difference between acute and chronic stress. So there's lots of evidence that short-term acute stress, and what I mean by that is where short-term stresses, or where what we find can actually enhance certain aspects of immune function, and it's all part of it's preparing our body to deal with the threat. So short-term acute stresses can actually be good for immune system function, and you can think of this in terms of it's basically arming the system, getting the system on high alert. But where we do really see issues is these longer-term chronic stress situations, which very much have the opposite effect, which is a suppressive effect on immune system function. And I just thought I'd show you, this is a where we've looked at lots of different procedures that livestock producers, our animals are exposed to, and what sort of levels of stress, pain, etc. we expect the animals to incur as part of those procedures. So as you can see there, handling, obviously some level of stress. The big ones are castration, mulesing, as you would expect. High level of stress, high level of pain. Weaning's another one, obviously not level of pain, but is quite stressful for animals. Whereas any of our activities like, ear marking, ear tagging, actual vaccination itself or drenching generally cause a minimum level of stress. It's these more chronic stressors like castration, mulesing, or weaning that we need to be a little bit careful about. And as producers, obviously, we often are looking to vaccinate at the same time as we're conducting these other procedures, because obviously labor costs are a major component for all livestock producers. So by doing multiple things while we've got the animals in the yards makes sense from that perspective. But we do need to understand that there are some consequences. And I'll just wanted to show you a graph. This is comparing antibody responses in lambs. So this is some work we did in my lab a few years ago. where we took lambs and we either we vaccinated them and put them straight back with mum, or we vaccinated them and marked them at the same time. And you can see the very clear reduction in antibody response we see when the animals were marked at the same time they were vaccinated. So we've done similar work looking at weaning. We see a similar response, not to the same extent as we see here, but it's just important for producers to be aware, although there's labour cost savings, etc., of us vaccinating when we're doing other husbandry procedures, there is a consequence of doing that as well. So just a little bit on macronutrients. So here we're talking about, the substrates that are necessary for our bodies. And that's the same whether we're talking about growth, immune system function. So we're talking about macronutrients like energy, amino acids, and other nutrients. And there's a lot of evidence demonstrating that that deficiency in macronutrients or insufficient nutrition has impacts on cell proliferation, antibody production, and also secretion of things like acute phase proteins by the liver. So animals that are nutritionally stressed will have a reduced capacity to respond to a vaccine. So I just put a paper here talking about periparturient immunosuppression. So what we're talking about there, periparturient, is the immune suppression we often see in animals around the time that they give birth. And especially just in dairy cows, where obviously, going into milk production has a huge, takes a huge nutritional resource for the animal to produce those large volumes of milk early in lactation. But it's very well known that the periparturient period is where we see immune suppression in animals. So, if we're vaccinating animals when they're immune suppressed, we can't expect the animals to respond to the same level as we would if they're not under that nutritional stress. So micronutrients, some of the trace minerals we know are really important for immune responses, and they include selenium, zinc, copper, chromium. We've already talked about chromium. And two different types of deficiency we need to consider here, primary or secondary. And when we talk about primary deficiencies, we're really talking about a deficiency resulting from a lack of consumption of that trace mineral itself. But we also see secondary deficiencies where it's due to a consumption of an element that antagonizes absorption of that trace mineral. So where animals are consuming an element that impacts our ability to absorb another essential trace mineral. So it can get a little bit complicated here, but there's lots of products out there now that, some of the trace element products that are really important and really beneficial for promoting good immune system function, and they will obviously, as a consequence, help with responses to vaccination. So just more specifically, selenium. We know selenium is really important for optimal neutrophil function. So neutrophils are a type of white blood cell that are really important in that innate type response. And we see this especially regarding mastitis in dairy cattle, especially during that periparturient period where they're immune suppressed. Often that neutrophil function is compromised, and we see increase in different diseases, including mastitis. Deficiency, we know, is associated with reduced B cell responses. Now, remember, those B cells are the ones that are producing antibody from that diagram we looked at before. Zinc deficiency associated with reduced B cell response, antibody production. You see some common themes here. Copper deficiency in B cell numbers. So all of these, a deficiency in any one of those will result in reduced antibody responses to vaccination. And chromium, we've talked about chromium from looking at its potential to reduce cortisol levels, cortisol induced by stress, as we talked about earlier. But we also know that supplementation with chromium is very effective in increasing antibody concentrations in the blood. So I just wanted to talk a little bit about genetics because it's something we often forget, and it's something that I've spent a lot of time looking at during my research career. So I just wanted to show you, so the diagram on the left is looking at antibodies responses to tetanus toxoid. So remember, tetanus toxoid is one of the components in your clostridial vaccines. And in each column, the dots there represent the response of individual animals. So along the bottom axis, the ones are actually a property. So 1H and 1K are done on the same property, and the letters represent a different year. So 1H would be a certain year, and 1K would be the following year on that same property. So you can, but I guess what I wanted to show you is the variability in antibody response you see in animals in your herds and flocks. So in this example, the animals in a given column on a given property have been raised together basically since birth. So their exposure to pathogens during their life is very similar. So here we're starting to really see what the genetic component of antibody responses starts to look like. So we've done a lot of work in this area over the years, and the ability of an animal, of a calf or a lamb, to respond, to generate an antibody response to vaccination definitely has a genetic component to it. And the heritability of those traits we know are about 0.3 or 0.4, which means about 30 to 40% of the variation we see in these responses we can attribute to the genetics of the animal. And that's very similar to a lot of other traits we see, like WEC in sheep, for example, would have a similar heritability. So I guess what I'm trying to demonstrate here is that in any given herd or flock, there will be some animals that will just be low responders. And that is not the fault of the vaccine, that is the fault of the animal. So on the left we're looking at tetanus toxoid. On the right, we're looking at responses in sheep to Virbac. And you see, again, we looked at a group of wethers, 2017 drop wethers, and then we looked at another group of wethers born the next year and looked at their responses to Virbac. And you can see between that, the highest response is over 100 times what we see in animals with the lowest response. So it's just important to realize for any vaccine, there will be an element of animals that are what we call non-responders. And that's generally not the fault of the vaccine. And we know that because they tend to be the same animals. So we've looked at responses to tetanus toxoid in sheep and looked at responses to Virbac in those same animals, and the low responders tend to be low responders to both. So just really, I thought it's really just important to have a look at the variability in response we see in animals. And somewhere on those graphs will be a protection level, and I'll guarantee that some animals are sitting under those protection levels. So we'll just start the talk now to sort of finish off about best practice vaccination. A lot of these things you'll be very familiar with. This is just taken straight from the Virbac site. So obviously the manufacturers of vaccine put a lot of effort into making sure when you receive the vaccine that it's sterile, safe to use. So it's really important that we do our part of the process, ensuring that the vaccines we're injecting into animals are not contaminated as best we can. So it's really important to sterilize as best you can your vaccination guns. The draw-off tubes, as we know, generally come with the vaccine, so that's okay. We can, different strategies, just boiling vaccination guns, probably not a good idea with plastic ones, but certainly with the metal vaccinating guns, you can boil them. Very good, easy way to sterilize them. It's important, though, to avoid use of strong disinfectants and soaps, and that's mainly because they can interfere with vaccines coming through those guns. We want to maintain cleanliness. Some of these things are pretty obvious. Needles sharp and clean. Now, this is a really important one. Replace frequently. I know there'd be very few producers who would replace a needle every 20 to 30 animals, but I would ask that you at least replace a needle every time you change a pack. I often see producers might do 500 animals with a single needle. That is not a particularly good idea. Use the shortest needles possible. So for sheep, 1/4-inch needles, this is obviously for subcutaneous vaccination. For cattle, 1/2-inch needles are ideal. Injecting high on the neck, we'll talk about that more specifically in a moment. Obviously for subcutaneous, under the skin. A really important one is to try and avoid injecting animals while they're wet. And that is because when animals are wet, there's a much higher probability when you administer those vaccines that you will drag bacteria, etc., with the needle into the injection site. So that's a real risk in wet animals, and it's also a risk in dusty conditions. So if you're in really dusty set of yards, it's a really good idea to try and wet the yards down, and of course always look at the product leaflet. So just more specifically talking about subcutaneous injections, it's important to, when you put the needle on your vaccinating gun, that we point the beveled edge of the needle away from you effectively. So if you look at the diagram on the right, what we want when we inject through the skin, we want that beveled edge to be sitting parallel with the muscle layer. And why that's important is it minimizes our chances of either injecting that vaccine into the muscle or intradermally into the skin. Generally, if animals can be restrained, I think it's ideal, and because you can use a two-handed technique. And what I mean by that is very effective way to ensure subcutaneous vaccination is actually to pull the loose skin up with one hand and inject. So we make a tent with the skin and inject with your free hand under the skin. So a good tip for subcutaneous vaccination, there should be no resistance felt when you're injecting subcutaneously. If you need to apply any sort of pressure to deliver that vaccine, you are most likely either in the skin or in the muscle. If you are in that subcutaneous space, you should feel no resistance when you inject that vaccine. Obviously, sometimes we can't restrain all animals, and particularly with sheep up a race, we can't. So we can do a one-handed technique. Really important in this case that when we vaccinate, we're coming in at a 45-degree angle. And if we come in perpendicular to the skin, our chances of going through that subcutaneous tissue and into the muscle is much increased. One thing we often sort of forget about too, which I thought is a really important point to raise here, especially with clostridial vaccines that contain whole cells, is mixing vaccines regularly. You'll be very surprised with a clostridial vaccine, if you sit that vaccine and it's not agitated, how quickly the cells will settle out in the vaccine. So the worst thing you can do when you're marking lambs is to hang up your clostridial vaccine on the top of your lamb marking cradle and not mix it. When you're vaccinating animals up and down a race, you're moving, so that vaccine does mix. But I always suggest to people to regularly invert that vaccine during the vaccination procedure. And that's really important to make sure that all animals are getting sufficient dose of all of those components of the vaccine. And that's especially relevant for whole cell vaccines that contain whole cells. So different routes of administration for different vaccines. Of course, today we're talking about sheep and cattle. So for all cases pretty well, it's a subcutaneous vaccine if it's injectable. But in other species like pigs, subcutaneous vaccination is quite difficult. So they generally use intramuscular, but these days they try and use intradermal. Now, ID being intradermal is where the vaccine's actually pushed into the skin because they're trying to get away from the need to intramuscular. administration because of issues with lesions, and we'll talk about lesions in a minute. But the diagrams on the right show you if we're using clostridial vaccines, any vaccine that's to be delivered subcutaneously, that's the area in that triangle where we should be targeting. And you can see the diagrams on the right, they're talking about the orientation of those needles. Some of our veterinary vaccines are intranasal sprays. A good example of that one's Rhinogard, the BRD vaccine. I'm not aware of too many oral vaccines for livestock. I don't think there is any, but certainly for humans, for kids, there's lots of oral vaccines. So I just wanted to flag that there are different types of administration, routes of administration for different vaccines. A little bit about transport and storage. Again, I've just taken this off the Virbac site. Very important to keep vaccines refrigerated. Super important not to freeze a vaccine. If you freeze a vaccine, I'll nearly guarantee that vaccine will be no good. So if your freezer plays up and your vaccine gets frozen, my very strong suggestion is you do not use that vaccine. It's also really important to protect vaccines from light. Some of the vaccine components can be degraded by light. That's the reason they come in a box. So obviously in the fridge where there's no light, that's okay, but try and keep them out of direct sunlight and use by the expiry date. Breaching new vaccine packs. So all different vaccines will tell you in what time frame should you use that vaccine once you first breach the pack. For the Webster suggestion for their clostridials to use it within 50 days. But every vaccine, all the manufacturers will recommend a time period. So I often get the question, what do I do if I just need a few doses out of a new pack and I'm going to put that pack away then for a period of time? So my suggestion in that case is definitely not just to pierce a new pack and start vaccinating and then throw it in the fridge with the gun still attached and pull it out 6 months later. My suggestion to people is go to your vet, get some sterile syringes and needles, just buy some alcohol swabs. And if you take your new pack of vaccine, you know roughly how much you need to finish your vaccination job. Very important to mix the vaccine before you draw anything out of it. Wipe the septum of that vaccine with an alcohol wipe and let it dry, let that alcohol evaporate and dry. Draw out with your sterile needle and syringe the amount you need and inject that into the pack you're using. And then that way, that keeps that pack basically sterile and effectively that'll be okay to store. But you've really got to take the time to make sure you do this, because contamination of those vaccine packs, as you'll see in the next few slides, is a real issue for vaccination lesions. So on to vaccination lesions, what are they? We've all seen vaccination lesions where you'll see a lump on the side of an animal's neck. If you've ever lanced one open with a scalpel, you'll see they're often full of really disgusting yellow pus. So vaccination lesions are really scars or abscesses at the vaccine injection site. So what causes them? So vaccine reactions are normal with some vaccines. Guadair is a good example, very potent vaccine. It's very common to get a vaccine reaction at the site of injection. But the important thing is they're usually transient and they will go away. What happens if generally the cause of those longer lasting vaccination lesions, they're generally caused by incorrect vaccination technique, poor hygiene or contaminated vaccine. So you think of a scenario where you're using an old needle, you've vaccinated 500 animals, they're wet, you're dragging bacteria into that injection site. The immune system tries to encapsulate that infection, and that's what causes this vaccination lesion. And it will sit there for quite a while. So, I often see people, they'll finish one vaccination pack, and before they put another one on the end of their drawing tube, they'll actually use the needle they've just vaccinated 200 animals with to punch a hole in the middle of the septum to give the new drawing tube a pilot hole. Worst possible thing we can do for vaccine contamination. If you want to make a pilot hole, get a sterile needle and do it with that, because what you're doing is basically just putting bacteria straight into your clean, sterile vaccine. So how to avoid the real key things? Don't vaccinate wet animals. Try to avoid dust in the yard. We've talked about these things. Use clean, sterile needles. Change them regularly. Ensure you're administering them correctly. So if your vaccines to be delivered subcutaneously, we've got to really work hard to avoid intramuscular or intradermal, and because that's a cost to you as a producer. When those animals arrive at the abattoirs, they will trim that lesion out, and you will lose that from your carcass weight as those animals go through. So it's costing you money. Avoid, and the big ones, avoid using contaminated vaccines by keeping them clean. We've talked about cleaning guns and avoiding the use of strong disinfectants. So just a couple more slides. Vaccine stability, we've touched on this. We have to be really careful with vaccine stability when we're using live attenuated vaccines, because the pathogens are live. Part of the reason they work so well is because they're live. If stability conditions aren't maintained, those pathogens will no longer be live, and so that significantly impacts the efficacy of the vaccine. So as we go from live attenuated down to subunit, the stability increases as we move away from live vaccines down to subunit vaccines. Things that affect it is obviously the type of vaccine, the presence or absence of preservatives. Now, this is an important one. Different vaccines will have different preservatives in them that are in there to stop bacterial growth. Some vaccines, for example, Virbac, have no preservative in them at all, and that's why they suggest once you breach the pack, you need to use it straight away. So those preservatives are really important, whether they're in there or not. Temperature, obviously, hot and cold. Keep cool, avoid freezing. We've talked about light, and we've talked about contamination. So just the last slide to go through. I just wanted to talk a little bit about a couple of hot topics in the area of livestock vaccination. So one of them is autogenous vaccines, and when I say an autogenous vaccine, it's really a vaccine that's customized to a farm or a region. So some of our complex diseases that we have to contend with as livestock producers, a really good example is bovine respiratory disease, are caused by multiple pathogens in variable combinations. So bovine respiratory disease, we generally have a viral pathogen combined with a bacterial pathogen, and it's that combination of pathogens that lead to respiratory disease, and in lots of different combinations. So as you can imagine, when there's lots of different pathogens in different combinations causing disease, it's really hard to provide blanket protection with vaccines, because those vaccines need to be targeting every different combination, which is very hard to achieve. So autogenous vaccines, what happens is, and they're very common in feedlots for BRD, where a veterinarian will come along and actually collect samples from animals that are infected with BRD, and so therefore they're getting an idea of what are the common pathogens that are causing BRD at this time in this location, and they will generate vaccines growing up those bugs or those pathogens and use them in vaccines that are customized for that specific application. So I thought I'd just mention that one. That's one that we're seeing more of out in livestock systems. The other one we hear a lot about is, or obviously there's a lot of news and a lot of worry about exotic diseases coming into Australia. Australia's certainly got a very clean and green image, and we need to maintain that. So obviously a way to protect against exotic diseases is to have vaccination programs. But one of the difficult things is, for many vaccines, once you've vaccinated an animal, it's very hard to differentiate whether that animal has seen the infection or has just been vaccinated. So, because often they will result in the same outcome in a diagnostic test. So there's a lot of work going on with vaccines, which we call DIVA vaccines, and they're vaccines where you can vaccinate an animal, but there's potential to differentiate between an animal that's seen a real infection versus an animal that's only been vaccinated. And that's really important when we think about exotic diseases and eradication program and diseases where eradication programs are in place, because that's very important for us to know, has this animal actually seen the disease, or is the results we're seeing just because it's been vaccinated? So I just thought I'd mention those couple of topics that are really on the top of the agenda for vaccines in livestock at the moment. So that's about it from me. Thank you very much for listening, and very happy to answer any questions you might have.